Many people recognize the Petri dish as the universal system for bacterial and cell culture. And this association is rightfully made, as this 2D system of growing cells has been the accepted method for cell and tissue culture for almost 100 years. But with new developments in cell culture, standard 2D cell culture is now not the only way to study cells and biological systems. I have been involved in undergraduate research at Purdue for almost 3 years now, in which I complete experiments using cell culture, so new advancements and methods in this area are very interesting to me and vital to my research. Today I'm going to talk to you about the recent advances in cell culture, as the development of 3D culture methods, address advantages and disadvantages of both 2D and 3D methods, and discuss future research possibilities through 3D culture, including drug discovery and organ engineering. As I mentioned before, the most common cell culture method today is 2D culture, in which cells are grown in a flat petri dish or a flask. As the cells grow in 2D, they grow as a monolayer, attached to the plastic surface of the flask or the petri dish. An article published in the Physiology Journal in 2017 by Duval et al. Explains the advantages of 2D culture. As the monolayer is formed in 2D culture, it allows for the access to similar amount of nutrients and growth factors present in the medium, which results in homogeneous growth and proliferation among all the cells on the plate. One of the most important advantages of 2D culture, though, is in its simple, efficient, and low-cost maintenance of the cells. Unfortunately, as described in an article in the Archives of Medical Sciences journal published in 2018, 2D culture also has numerous disadvantages. 2D culture does not accurately mimic the conditions of cells in vivo, and it lacks the same cell-to-cell and cell-extracellular environment interactions that are present within the body. These changes from the body to 2D culture, or the lack of these interactions in 2D culture, can change many aspects of cell differentiation and function, which then makes 2D culture not a reliable source for studying in vivo systems. 3D culture, on the other hand, is a relatively new way to culture cells that includes a culture environment that allows cells to proliferate and interact with their surroundings and extracellular matrix, or other cells, in 3 dimensions. Jacob and Young published an article in Frontiers in Molecular Biosciences in 2020 in which they discussed 3D culture methods. They found that the presence of the extracellular matrix and the surrounding cellular microenvironment in 3D culture significantly impacts cell proliferation, differentiation, and other cell survival methods. 3D cell culture can also be used in a way that replicates in vivo cell environments, so it can be used to study in vivo cellular processes more accurately than it can 2D. Some of the areas of study that have benefited the most from the use of 3D cell culture includes the areas of drug discovery, organ generation, and the general study of healthy or disease states and tissues within the body. As long as the microenvironmental factors are consistent with those in vivo, 3D culture has enabled real-time and a more accurate study of tissues in both their healthy and disease states to answer the basic fundamental questions in biological systems. The accuracy in modeling in vivo systems makes 3D culture valuable for testing the effects of drug development and drug treatment. Imamir et al. Published an article in 2015 in which they compared 2D and 3D models as drug testing platforms, and they found that there were many differences between the two models in terms of cellular effects after drug treatment. Most importantly, they found that 3D culture provided results most similar to the in vivo experiments they performed, showing that 3D culture produces results more similar to in vivo systems in drug testing and development. And lastly, with the ability to accurately model in vivo environments, it becomes possible to engineer complex tissues and organs outside of the body, specifically using patient cell samples, as these cell samples can be taken from patients and grown in 3D culture to create compatible organs for transplant, or to test drug treatments to see how the cells inside the body would react. Though 3D cell culture represents an exciting new way to research cellular functions, disease, and therapeutics, it is still not as widely used as 2D culture. Sun et al. Published an article in Advanced Nanobiomed Research in early 2021 in which they explained that 3D culture is still used less than 2D in general due to its complexity and high cost of materials. They argued that once 3D culture can be more accessible to labs everywhere, it would have to be a standardized protocol and optimized materials production, and then everyone will be able to use it. With the development of 3D culture alongside 2D methods, researchers now have the ability to accurately study in vivo cell function outside of the body. This opens many new exciting fields for in vivo research, including the study of healthy and diseased tissue function, drug discovery and therapeutics, and patient-derived organ engineering. As both 2D and 3D culture continue to become optimized and develop for a larger variety of cellular systems, our understanding of tissue function, diseases, and cancer will continue to grow.